ครับสวัสดีครับ Hi everyone. Today is actually Wednesday, the 1st of April. Actually, today it's an April Fool Day, but unfortunately, this is the real things I would like to say. This afternoon, I would be able to conduct the class, so that uh, I decided to uh, do the video recording so that you can study these video clips to do the lab reports. Actually, this day we are going to conduct the labs 7, which will be about the nitrogen determination. Okay, so let us first uh, get into the backgrounds of this lab. First of all, I'd like to go back to our uh, presentations. Uh, in the lectures, if you still remember on the first chapter, in this chapters, almost the last slide, or actually it's the slide number 60. We had talked about the domestic wastewater characteristic. Several parameters have been used to describe the characteristic of the wastewater. Um, several characteristics of several parameters have been used to describe the characteristic of the domestic wastewater. For example, the total suspended solids, the BOD5, which is very important in terms of the organic matters, the determinations in water sample, especially the wastewater, because domestic wastewater, the major pollutants is basically the organic substance, and we generally measure in terms of the BOD, biochemical oxygen demand, or the COD, chemical oxygen demand. So these two are the most common parameters which we had described and we had discussed last week. And the next parameters is the ammonia and nitrogens. These parameters we will talk about today, and also the organic nitrogens. Organic nitrogens, both actually, I can say that total nitrogens. Combinations of these two are basically the total nitrogens found in the domestic wastewater. In general, we call it TKN. TKN actually stands for the total gel down in nitrogens. Nitrogens become one of the major uh, parameters or the major contaminants found in wastewater. And this actually comes from the organic substance itself. We all know that one of the major organic substance, which is protein, their monomers consist of the amines groups. Amines is the NH2 associated with other organic molecules. And once it breaks down, so it becomes an ammonia. So if it's associated with the organic molecules in proteins, it becomes the uh, polymers of the proteins, and this one we generally call an organic in nitrogens. But once it breaks down, we usually call the ammonia and nitrogens. Combinations of these two are quite common in the domestic wastewater. These two parameters are quite common in the domestic wastewater, so we generally find in a significant amounts along with the phosphorus. Phosphorus essentially comes from the laundering activity or the watching activity. Mostly the detergents we used essentially contain some amounts of the phosphorus. So once we used this phosphorus, we goes into the wastewater. So in the wastewater treatment plants, we normally monitor the ratios of the BOD is to N is to P. For the aerobic treatment, like we are using the microorganisms and providing the oxygens to degrade the organic matters. So this reaction would essentially need the nitrogens and phosphorus along with, but they only need in accordance with the ratios of about BOD is to N to P, 100 is to 5 is to 1. So if it's present in a greater amount, I mean, like a BOD is supposed to be 100 and nitrogen is supposed to be only 5 and phosphorus is supposed to be only 1. But if it presents in a greater amount, like in here, the BOD for the low strength wastewater, it is about 110. But if you consider the amounts of the nitrogens, the total nitrogens combinations of these two becomes 20. 8 plus 12 becomes 20, and the phosphorus is 4. Of course, according to the ratio BOD is to N is to P, it seems to be the ammonias and phosphorus will be left anywhere. So the leftover, the nitrogens and phosphorus will finally disposed along with the treated water into the natural receiving stream or the surface water sources. And this, we all know that the presence of the nitrogens and phosphorus will enhance the growth of the algae. Growth of the algae could be enhanced by these two nutrients. And if we keep discharging the treated waste containing nitrogens and phosphorus, our receiving water surface water sources will end up with the eutrophication problems. I hope you still remember these keywords, the eutrophications, which generally occur in the marsh or the lake receiving nitrogens and phosphorus. So these are the major pollutants found in the domestic wastewater, but it may not be found in some industrial wastewater. Oil and grease, of course, some certain amount of oil and grease will be there because they in our daily life, we have to cook, we have to eat, so the kitchen waste always contains some oil and grease. The other parameters, for example, the bacteria or the pathogens, mostly the pathogens that are typically found in the septic tank or the human excreta is the total coliform bacteria. But if we really find it in the human excreta, it is the fecal coliform bacteria. Fecal coliform bacteria, it's the groups of the coliform bacteria. So total, it's overall microorganisms overall the bacteria, which typically found in mammals, but the one which typically excreted from human being, it's a fecal coliform bacteria. So this is actually a subgroup of the total coliform bacteria. Normally we report it in terms of the numbers per 100 ml. It gives a sense like if you are taking hundreds ml of wastewater, you will typically find the coliform bacteria approximately 10 to the power of 6 to 10 to the power of 8 for the low strength wastewater. So this is like the number. the cells that can be found in the low strength wastewater. So Asaphicoliform bacteria is a subgroup of the coliform bacteria. The number that we normally find it's smaller than the total coliform bacteria. Okay, in both low strength and high strength wastewater. So low strength in this context normally refers to the gray water, which is the wastewater from the washing activity, showering, sink, anything except from water closets. The waste from water closets normally it's kept in the septic tank, and that could be called the black water. So we recognized the liquids in septic tanks as the black water. So this could be a high strength wastewater, high strength domestic wastewater. The BOD could be actually 350 to 400 milligrams per liter. This is quite common. But for the low strength gray water, the BOD could be around 100 or even less than 100 milligrams per liter. To remember that according to the effluent quality standards, we are allowed to discharge the BOD smaller than 20 milligrams per liter, not as high as 100 milligrams per liter like this. Other two are the helminth, cyst, Giardia lamblia. It's also the helminth, Cryptosporidium or cyst. This is also the helminth. So helminths are allowed to be found in a small amount with respect to 100 ml of the wastewater sample. So these are the characteristics of the domestic wastewater, which we will focus on after midterm examinations. So we are now, this week, we are going to do the ammonia nitrogens. Last week, we were doing the BOD analysis, BOD and COD. And then the next week, we will do the total suspended solids, which we will use the gravimetric method to measure the solids found in water sample. So okay, this is the what so-called the wastewater characteristic. So just come back to our today's lab. We will do the nitrogen. We will measure the amounts of the ammonia nitrogens. We will also measure the amounts of the organic nitrogens in terms of the TK and total gel down nitrogen. And just some just have a look some more remarks here. The units of the nitrogens, it's normally expressed as the milligrams per liter as nitrogens, not as the ammonia or not as the organic nitrogens, so that these two amounts can be combined as they are both having the same unit. Both are the expressed in terms of the milligrams per liter as N. It's already with respect to the molecular weight of the nitrogen, not with respect to the molecular weight of the ammonia or the organic nitrogen. So everything is expressed in terms of the milligrams per liter as N. So this value can be combined. So the conversion from the milligram per liter as the ammonia or the milligrams per liter as the organic nitrogens to the milligrams per liter as nitrogens. I have already explained it sometimes before the midterm exams. I couldn't remember. A long time ago, we had discussed about the unit conversion already. So, okay, this is the general backgrounds about the wastewater characteristics. So if you still remember the midterm exam question, there was one question about the turbidities and colors in wastewater. Actually, the turbidities and colors are not significant parameters describing the wastewater characteristics. Normally, we rather measure the total suspended solids, not the turbidity. Turbidity is only used. We use only for the identifying the surface water and also the tapped water characteristic. Okay, so just come back to the nitrogen. So it's a switch to this page, the OneNote. Hopefully you are still with me on the sharing screen. Okay, so this one, these files I have left this file in the files in the background folders, or you can download. Okay, so for the nitrogens present in the nature, before going into the details, I'd like to introduce you about the nitrogen cycle. This is the figures describing the nitrogen cycle in nature, which I obtained from Sawyer 2003. This textbook, actually, the title is Chemical Chemistry for Environmental Engineering and Science something. You can visit the libraries and have a look. It's there in the our faculty's library. So in this cycle, you will find in the nature there are various forms of the nitrogens. For example, ammonia, organic nitrogens. Sometimes it exists in terms of the nitrates, oxide. Sometimes it exists in forms of the nitrite, and sometimes it exists in the forms of nitrate. So it changed the forms regularly, commonly change, and sometimes it turn it back or change the status from the aqueous forms to their gaseous forms, becoming the nitrogen gas and existed in the air like that. So this is a very common forms of the nitrogens that are typically found in the nature. So if we focus on the nitrogen forms which commonly found in the nature, we could list it like this, and especially in a water and wastewater sample. We normally find ammonia, nitrogen gas, nitrite, and of course the nitrate. So nitrate is the most oxidizing forms of the nitrogens found in the nature. And ammonia seems to be the most reducing forms. Why I'm saying like that? If you are take if you take a look at the oxidation number, you will find the oxidation number of the nitrogens in the ammonia becomes -3. Hydrogen is +1, so there are 3 moles of the hydrogens associated in the ammonia's molecules, so that ammonia becomes a -3. Nitrogen gas is the gas, of course, the oxidation number is the 0. And if they associated with oxygens, 2 moles of oxygens are associated with the nitrogens becomes a nitrite. So this one, if you really take a look at the oxidation number, so you will find it's +3. Actually, this one, 1 mole is -2, but there are 2 moles of the oxygens, and the charge is minus, the overall charge is minus, so that the oxidation number of the nitrogens is +3. And for the nitrate, this one is called nitrite. For the nitrate, it's actually +5. This one is -6. Overall charge is -1, so that the oxidation number of the nitrogens in here is +5. Actually, there are many forms of the nitrogens, but these are the 3 to 4 forms that commonly found in water sample. So they can actually change the forms. They can actually change the fate and the status. For example, the nitrate in the nature, by the photosynthesis, nitrates can be utilized by plants to synthesize the proteins, while the carbon dioxide is also used some of the biological reaction conducted by bacteria are called the rhizobium, or actually rhizobium is more or less is a fungi, but some bacteria also can conduct this reaction. So the nitrogen gas in the air can be fixed. These reactions is we call the nitrogen fixations. So these bacteria can actually catch the nitrogens in the air and turn them into the proteins. Similar to the ammonia, similar to the nitrates, ammonia can also be used by the plants during the photosynthesis to synthesize the proteins. So as here you can see that the proteins are forms. These proteins we normally call the organic nitrogens. Okay, and for the proteins, on the other hand, the microorganisms can also change these proteins into the ammonia, so that ammonia actually it's the breakdown of the nitrogen ammonia molecules amines from the amino acids. So ammonia it's actually separated from the proteins of the organic itself. So this ammonia can be oxidized by oxygens. Under the presence of the oxygens, ammonia get converted to the nitrite by the nitrosomonast, and the nitrites can be further oxidized by the nitrobacter, another group of the microorganisms, and nitrites will be converted to the nitrate. Combinations of these two reactions, actually these are the series reaction. First reactions will take place and then followed by the second reaction. Nitrite is more or less the intermediate. Normally under the oxidizing condition, nitrite is unstable forms of the nitrogen. Ultimately, it has to be converted to the nitrate, which is the most stable forms of the nitrogens under the presence of the oxygens or the oxidizing condition. So these two reactions typically called the nitrification. That is why in this nitrogen cycle, you find lots of many terms of the many nitrification used to describe the reactions. Here there is also nitrification. Here is also nitrification. Here is also nitrifications. Nitrification is something ammonia or the nitrites get converted to the most stable forms of nitrogens, which is the nitrate in the nature. So this one is called the nitrifications. Normally, the wastewater contains some ammonia and some organic matters. These organic matters will be broken down to become ammonia first, and then ammonia get converted into the nitrate. This usually happens in the bioreactor, which we provide the oxygens. But anyway, if we are talking about the nitrogen removal, if we are not completely remove the nitrogens from water, still it is there, even it just converted into the nitrate. But anyway, the nitrogen is still there. So if we want to completely remove the nitrogens from wastewater, so how can we do? We have to let them react with the organic substance, and then this nitrites or nitrate get converted into the nitrogen gas. So this reaction will only occur when the oxygen is absent, so that nitrites or nitrates becomes the electron acceptor instead of the oxygens. Okay, so these reactions will occur only under the absence oxygens in the water, or in general, we call this condition as the anoxic conditions. Anoxic condition, I'm not sure whether you have ever heard it about anoxic condition. So if the oxygen presents the degradation like this, we normally call it aerobic degradations or aerobic oxidations. Uh, this will occur only under the aerobic conditions, but this will occur only under the absence of the oxygens, or we typically call it anoxic conditions. And this reaction is uh, called denitrification. So we have nitrifications and following and the conversions of the nitrates to the nitrogen gas. This one we call the denitrification. That is why in the nitrogen cycle, so if the nitrate get back get converted back to the nitrites, and nitrites become a nitrogen gas or the N2O. These reactions are typically called denitrifications. Okay, so these are the reactions generally found in the nature, especially in water and wastewater treatment. Now, just come back to our raw waste. In the raw wastewater, in a raw waste water, it essentially contains some organic nitrogens and ammonia. So once the waste is just discharged, we usually call it raw waste, or just generated, or becomes just become a wastewater. This kind of wastewater we call it raw waste. For raw waste, you can expect it to find high amounts of the organic nitrogens. So you just use that water, and that water becomes a wastewater. So in the wastewater, there are a lot of organic substance get along with, and that organic substance, some are proteins. So proteins essentially contains some nitrogen, and this we normally call the organic nitrogens. So you can expect high amounts of the organic nitrogens in the raw waste, while the ammonia presents in a small amount. But if you keep this wastewater for a long time, like several hours to several days, these organic nitrogens will be broken down to becomes ammonia. According to this reaction, proteins by the bacteria, the breakdown of the organic substance will take place, and these nitrogens become ammonia. So at the end, you will find a significance of the ammonia at the end after we keep wastewater for a long time. But for the raw waste, you will find a significance of amounts of the organic nitrogens and not the ammonia. So it just reverts when we keep it the wastewater for a long time. But anyway, both a combination of organic organic nitrogens with the ammonia are called the TKN. So TKN actually stands for the total gel down nitrogens. Okay, so this normally called the TKN, total gel down nitrogens. So according to the reactions taking place in the nature, the organic substance, if you really take a look at the organic substance, organic nitrogens, the organic nitrogens will get degraded and become the ammonia. So if you are focusing on the amounts of the organic nitrogens present in the water sample, you will find, you will see some reductions like this. So this reductions is actually following the first order kinetic. But if you have a look on the ammonia, you'll find the ammonia will gradually increase as the organic substance break down. The amounts of ammonia will gradually increase until reaching the maximum value. So after it's reached the maximum value, so it gets again a reduction. Ammonia somehow get converted into the nitrites and becomes nitrates according to the nitrification reaction. That is why sometimes the nitrites, the amounts of nitrites were increased, and later on drops to almost nearly zero, while the nitrates were increased at the end. So this actually was due to the nitrification reactions. Okay, that is why if you are forecasting the change occurring in forms of nitrogens in the polluted waters or in the wastewater samples under the aerobic condition, you will find a concentration profile like this. Reductions of the organic substance, increase of the ammonia at the initial stage, and at the end, both ammonia with nitrites will reduced, and at the end, the nitrates will form. So at the end, you will find significant amounts of nitrates in the treated water. So this is also these figures I also obtained from the Sawyer 2003, the same textbook that I have taken the nitrogen cycle. So these are the background information about the organic nitrogen, ammonia, nitrite, and the nitrate. But there's something important about the ammonia. So if it presents in the nature, so what will happen? In fact, if we are talking about the ammonia in water, ammonia in water can actually exist in two forms, both in both the NH3 forms and NH4 plus forms. So these two are both ammonia, but this one is the dissociated forms. Ammonia NH4 plus. This one is the dissociated forms. They are having a very good interaction with other anion. They have great solubility, great capable for the solubilities in water. So they actually completely dissolve in water and have ability to interact with other ion, positive cation or anion, negative charge ion. But sometimes they also exist in this form, so undissociated forms. This is called the undissociated forms. This may be the forms of ammonia that we are familiar with, but maybe not this one. But in fact, it depends on the pH, whether it exists in this form or this form. If you really take a look at this equilibrium, ammonia plus the hydrogen ion and becomes the NH4 plus. If the pH is getting increased, pH higher pH means this amount hydrogen ion gets lower. So if this one gets lower, what will happen? The equilibrium will shift to this size. So according to the equilibrium principle, the reactions will shift to this size to make it equilibrium second. Because it's unbalanced, this one it gets small so that the equilibrium will shift to the left side. That is why more ammonia we express in terms of the NH3, or we can say that the NH3 is predominant. So if the pH is higher, but if it's getting lower, lower means concentrations of the hydrogen ion is high, then the equilibrium will shift to the right side of the equation. Therefore, the ammonium ion will predominate under the low pH. So the ammonia, which form of ammonia will exist, is greatly depending on the pH value. But anyway, if it exists in the forms of the ammonia and H3, this form is actually more toxic to the microorganisms and also the living organisms in the water or the aquatic organisms. As this forms the ammonia, NH3 can freely penetrate to the microbial cells. But concerning about the this one, NH4 plus, as they are the dissociated forms, as it is the dissociated form, so uh, there is a charge around. That is why it's a bit difficult to diffuse through the membrane of the cells. So that is why we can say that talking about the toxicity to the microorganisms. So which one is more toxic? NH3 is more toxic. Okay, and it depends on the pH. So the greater pH will be the greater concentrations of the ammonia NH3 found in water sample. So this one show the total ammonia nitrogens both exist in NH3 forms and NH4 forms. So this is just a combination of these two is starting from 0.1 to 10 milligrams per liter. But at any concentration, no matter it's low or high, the higher pH will be the higher concentrations of NH3 exist. NH3 forms exist. So you can have a look at the relationships shown that in this table, in this figure. But anyway, in the analysis of the ammonia and the nitrogens in the wastewater, for the ammonia, as I told you, ammonia can actually exist in two forms, NH4 plus and NH3. Combination of these two is called the total ammonia and nitrogens, but it depends on the pH, whether it will present in the NH3 forms or in the NH4 plus forms. But anyway, in the analysis of the ammonia, in the measurements of the ammonia, normally we can measure it directly by using the distillation techniques. So under the higher temperatures, the ammonia will be stripped or escaped from water. That is why we can use the distillation process to separate the ammonia from water. But there are some constraints. Ammonia will be escaped only when the pH is greater than 8. When the pH is greater than 8, okay? Normally we maintain the pH as 9.5, like that. So if the pH is getting higher, this form of ammonia will present NH3, and this form is easily escaping as compared to the NH4 plus. So we maintain a high pH because we want the ammonia exist in the this forms, not in the this form. This form is difficult to vaporize because it is the dissociated forms, so it can actually interact or associated with other anion. That is why it's a bit difficult to escape from the water sample. So we prefer the ammonia present in the NH3 forms while doing the analysis. So what we can do it with the ammonia analysis. So we use the distillation unit to allow the ammonia stripped from water sample. So we heat this water sample until the ammonia come up, and then get distilled, and before being collected in the flask containing boric acid solutions. So this flask, we normally put the boric acid inside, and this boric acid inside can actually absorb the ammonia. So what is the functions of the boric acids? The boric acids actually can actually change the ammonia come along with which come to the columns and before being distilled. So this ammonia will be absorbed by the boric acid. This is the boric acid H3Bl3. So once it absorbs, ammonia becomes the NH4 plus, and the boric acid becomes H2Bl3 minus. The presence of the boric acids generated due to this reaction can be measured by performing a titration of these solutions against the sulfuric acids, but its concentration is only 0.02 normal sulfuric acid. This one, titrations against this one, and at the end points, the colors will turn it from either green or blue to the brown endpoints. So you can have a look here. This is a figure showing the colors changing. So at the beginning, once the boric acid is absorbed, so if the concentrations of the ammonia is too high, the colors will change to green. But if the concentrations is not too high, the ammonia represents we change the colors of boric acids to blue or the dark blue. But anyway, if we perform the titrations by using mixed indicators as the indicators identifying the endpoints, so additions of the sulfuric acid 0.0 to normals of the sulfuric acids, when it reached the endpoints, the colors will change to brown colors like this. This is the initial, and this is the after doing the titrations for some time, and the last one is the water sample once the endpoint is reached. Okay, so for the distillation unit, this is the what I have sketched. It's just the general. It's just the conventional distillation unit, but in fact, we will use the distillation unit like this. We will connect the tube containing a water sample. We connect this tube with this distillation machine, and then the columns is on the right here. In this columns, ammonia will be condensed and then flowing along with the liquids and before being absorbed by the boric acid solutions down here. Okay, so this one it takes like only 4 minutes to distill it. But if you use the conventional unit like this, it may take an hours or more than 1 hour. Could be 2 hours like that. It depends on the volumes of the water samples, and also it depends on the strength of the ammonia present in the water sample. We do the titrations, and then we also record the amounts of the sulfuric acid used, and then this one will be used for the calculation. These are the procedures that we measure the ammonia. But for the organic nitrogens cannot be measured directly. Okay, it cannot be measured directly. It must be digested. Okay, it must be digested and converted to the ammonia first. In a wastewater sample, as I told you, there will be a combination of the organic substance and the ammonia. So raw waste, significance of the organic nitrogens is found, and then we do the digestion process. The digestion process will convert the organic nitrogens into ammonia, and at the end we measure the ammonia in water sample ammonia that we determine from this step will be the ammonia which already exists in combination with the ammonia which get converted from the organic substance. So the combination of these two, if you measure by using the same techniques, you will find that combination of these two is called the TKN. TKN is equal to organic nitrogen plus the ammonia in nitrogen. Okay, so the organic nitrogen is equal to the ammonia, TKN minus the ammonia. So these two we obtained directly from the analysis. So these two we obtained directly from the analysis, both TKN and ammonia. So after the digestion process is finished, you can begin it with the distillation process to determine the ammonia. Okay. These are the steps. So digestion steps. So we normally put the sample, 100 ml sample into the TKN tube. This tube is actually the borosilicate tubes. It can withstand with the temperatures over 500 degrees Celsius. We put the water sample, 100 ml, and then we put the copper sulfate. Copper is used as the catalyst, and then we put 6.7 ml of the sulfuric acid, and we also put the potassium sulfate for the digestion. So this one, we put it like this. Then we connect this tube in the digestion unit. So this is a photoglass of the digestion unit. Luckily, you are not doing this labs this week, because if you are doing this lab, if you are going to do this lab, it will take like 3 hours. So it's look like this, the TKN tube. So once we completed the digestion, all the liquids is gone or become vaporized. Only a small amount of liquids present, clear, but you may be seen like a blue, brighter blue colors solutions left at the bottom of the chamber. So once we leave it like this, and the next step is we have to add the distilled water and of course the sodium hydroxide, because during the digestion we prefer to distill it, the sample under the pH above 8 or at 9.5. That is why before doing the distillation, so we have to add some certain amount of sodium hydroxide so that the pH is greater than 8. That is why the ammonia can be stripped efficiently if we could actually maintain the pH greater than 8, or we can actually maintain approximately 9.5. So the distillation process will only take 4 minutes. 4 minutes. Everything will be complete. Only 4 minutes. So the receiving solution is the boric acid. So we put around 25 ml of the boric acid here. We used as a receiving solutions, and also we add the indicators, few drops of the indicators, a few drops. Sometimes it means 3 drops is enough, sometimes it means 5 drops, but anyway it depends on the condition, so you can decide by yourself how many drops you require for the mix indicator. So this mix indicator, so once you add it into the distilled water, so the colors return green. But after we prepared everything, so we finish, then we put it here. during the titration. So what will be the colors of the solutions after the titrations? So the colors will change from yellow which colors to dark blue and probably to green. Okay, so at the end we do the titrations, the colors return back again from blue to green, from green to bright brown. So these are methods that we use to measure the TKN, okay, TKN and ammonia. And if we know these two forms, we will get the uh, organic nitrogens accordingly. So these are the background information about the nitrogen determinations, its reactions, and how we can conduct these labs. For the nitrogen's determination, you can simply have a look these files, determinations of the total gel nitrogen. So you have to begin with the step 1, digestion. We need to add 3 different reactants into the water sample, and don't forget to add the glass bead. Glass bead will help to separate the heat or to distribute the heat eventually in the tubes. So if we don't put a glass bead, sometimes it gets boiled, and then the heat which accumulated get exposed. So if you open this, there is very high chance that all the hot liquids will come out. So just you just have to be careful if we don't put the glass bead. So putting the glass bead would be better to distribute the heat eventually in the entire tube. So we have to digest under the hood and until the liquids, the amounts of liquids reduced to only few millimeters stick to the bottoms, like the figures that I've just show you. And after that, we add some certain amount of sodium hydroxide to bring the pH to be greater than 9.5. And then do the titrations. Sorry, distillations. Okay, just to distill it, the ammonia. And at the end, we have to do the titrations against the 0.02 sulfuric acids. In this case, you have to do the blank. In this case, you have to do the blanks just to make a comparisons between the ammonia found in water sample and the ammonia typically found in the distilled water. That is why at the end you have to calculate the amounts of ammonia, which is equal to the amounts of the sulfuric acids used for samples minus the amounts of the sulfuric tritrate for the blank, multiplied by 280 divided by the ml samples, so you finally get the ammonia. For the ammonia, you only do the step 2 and the step 3, but for the TKN, you have to begin with the step 1, step 2, and followed by the last step, step 3, the titrations. Remember that the total gel nitrogen delta TKN is equal to the total organic nitrogens plus the ammonia and nitrogens. So this is the brief methodology about the TKNs and the nitrogen determination. Now, if you want some more information, you can go to the files, and in this files, in the folders class materials in background, you will find the my files, and also If you go to the protocol, you will get two relevant files, lab 7 and the one which I have just shown you. So in the presentation file, lab 7's presentations, this file, okay, so there are some certain explanation you can find out. There are some material required for the digestion, what we have to do. So it's all summarized here. We need to put a glass bead. We need to put 100 ml of sample. We have to add potassium sulfate, copper sulfates, as well as the sulfuric acids to allow them to react under the higher temperatures. In this box, it's inside there is the dry heater. The temperatures could move up to several hundred degrees Celsius. So it is very hot. That is why we have to be very careful. And during the digestion, what will happen as we put the 6.7 ml of the sulfuric acid here. So if we do the digestion, there is a chance of the fumes get escaped from this unit. And if we are not doing this in the hood, so just imagine all the acids fumes will affect the one who are doing the analysis. Inside, there are some procedures so that you can actually read it by yourself. Distillation unit is something different, but not the major difference. Titration, you have to titrate from pale green to the pale lavender and finally to the brown gray. Brown gray actually indicates the endpoints of the titration. And finally, you will have to calculate the amounts of ammonia nitrogen present. For the ammonia determination, as we haven't do the digestion, in this case, in order to increase the pH of the solution, so we put the borate buffer instead. 5 ml of borate buffer is quite enough to analyze the ammonia. And phenolphthalein is also added along with. Phenolphthalein is there, the pH is 8.3. So if we drops the phenolphthalein into the water samples for the ammonia determination, you will find the colors turn pink like this. The colors turn pink. If the pH is greater than 8.3, the phenolphthalein will turn to pink. But if the pH is smaller than 8.3, the phenolphthalein will express colorless solutions in the solutions. So these are the how we will do the TKNs and also the ammonia and nitrogen determination. You can also find out the organic nitrogens by taking the TKN subtracted by the ammonia. But what will happen if you are getting like the ammonia greater than the TKN? Theoretically speaking, TKN must be greater than the ammonia. It's impossible that the ammonia will be greater than the amounts of the TKN. Okay, so these are all about the analysis. Then we just have a look at the results that will be distributed to you by 3 o'clock today. The results will be something like this. A sample were collected from various sources influent to the wastewater treatment plant and also the effluents from the treatment plant. And the raw data will be given. The volumes of the sample used is 100 ml because sometimes we don't have to use the 100 ml. Sometimes we only use 50 ml or 20 ml, but we have to make up the volumes to 100 ml. In this case, at the end, you have to multiply it by the dilution factor once you calculate it to determine ammonia or the TKN value. The Biuret titrations results obtained from the titrations is shown in these two columns. You can take the stop minus the start to get the exact volume of the 0.02 sulfuric acids, so that you can use this information to determine both ammonia and the TKN, similar to the effluents. And one more thing, the blank titration. Blank titrations. Here it shows the starting points and the stopping points. As I told you, there's a chance that there might be some ammonia presence in the distilled water. So in this case, we have to correct the value, and also there must be some problem with the color development or some ammonia which may be there. That is why we have to do the blank analysis. So we have to make a comparisons with the blank. So there are only 2 pages for the results of this week. You can find out the TKN, and you can also find out the ammonia. I suppose I'm not sure whether which one is for ammonia and which one is for the TKN determinations, but according to the titrations, you have to make a decision which one is the ammonia, which one is the TKN. Ammonia must be smaller than the TKN. That is the typical value you should obtain from the experiment. Okay, so you are supposed to do this. So each group will get similar results like this, just to find out the ammonia and the TKN. And at the end, you can determine the organic nitrogens by taking the TKN minus the ammonia. I'm not sure whether which one. I'm not sure which one is the is for ammonia, which one is for the TKN. So you have to check the results again. I think one is the TKN and another one must be the ammonia. Okay. Okay, I think that is all for today's labs. So for the discussion, I think maybe next week we can do it together with the nitrogens. I mean the BOD and COD, the discussion, we can do it along with the nitrogen next week. So this week, so I have no more about the nitrogens. So if you have any inquiry regarding, again, you can check.